So I wanted to talk to you today about like genomics tools for interpreting patterns of somatic driver and passenger mutations in cancer. And if you work kind of like a little bit in cancer, you've probably seen this slide over and over again, like that we feel like why are driver mutations important is because we believe that cancer actually evolves based on like subsequent event of different driver mutations. And each of those driver mutations gives the cancer like some survival or proliferation advantage to proliferate over its normal tissue. But as you probably also know, this is just how the story, because with every driver mutation event, there's like a plenty of passenger mutations. Actually, most of the mutations that we see in cancer genomes are passenger mutations. So if you want to pinpoint those mutations in cancer genomes that are functionally important, it actually becomes like searching for a needle in a haystack, because you have your few driver mutations, but most of the mutations are passenger mutations. And you can basically approach this problem from two angles. Either you can try to characterize the passenger mutations, or you can first try to characterize the driver mutations. And either way, you'll actually arrive at a classification of driver versus passenger mutations. But I think it's important to keep both of these like components in mind, that they always go hand in hand if you want to have a reasonable detection of driver versus passenger mutations. So why are we actually interested in driver mutations at all? And I think there's like three major reasons why we are interested in cancer genomics in clinics. So the first reason is that we actually know about cancer genes that are important in tumor formations based on their driver mutations. So a lot of the genes we actually have only discovered, not necessarily experimentally, but by large-scale computational analysis, and to see like where mutations actually have accumulated. And they've told us a lot about like how cancer actually works. The second reason why we're interested in driver mutations is because driver mutations are diagnostically and therapeutically important. So knowing exactly about like which genes are involved in which tumor types gives you actually therapeutic opportunities for designing specific small molecule drugs specifically tailored to the individual patient's cancer genome. And the third reason why we're interested in driver mutations is because at this point, we have robust profiling platforms to sequence up to the entire genome of a cancer patient for leveraging these knowledge into clinical diagnostics. And I think this is pretty remarkable, given that 20 years ago, the Human Genome Project was just completed for one genome, and now we can basically sequence an entire patient genome just in clinical diagnostics. And I think it's really important to keep in mind that it is always this interplay of population variant discovery and optimizing the therapy for the individual patients that makes actually cancer genomics very characteristic and interesting. So on the one hand, you have these large patient cohorts from which you want to extract like those mutations in these biobank scale databases that are functionally important for cancer formation. So as I said, like searching for the needle in a haystack. But on the other hand, you really want to use these like large scale biobank scale data and actually focus them for the individual patient. And that is actually like how you identify the individual target lesion in a cancer genome, in a patient's cancer genome that you might leverage for a small molecule inhibitor therapy. And this is really where computational tools in cancer genomics come into play, and why I think they're like so important, not only for the interpretation of the biobank scale data, but also for like making clinical decisions for the individual patient. And usually we don't see a patient only at one single time point, but we usually see the patient over multiple time points over the disease. So usually like there's some type of sequencing involved in the initial diagnosis of the patient for characterizing the initial genomic profile of the patient and making the initial therapeutic decision. But unfortunately, after like the initial clinical response, most patients will eventually acquire some form of resistance. And this is where actually like you have a second profile usually from the cancer genomics platform to design the second line therapy. So it's usually like really a sequential follow-up over time where like cancer genomics analysis work together with pathologists and clinicians to make therapeutic decisions. So how do we identify driver mutations in cancer? And actually, this problem has been actually like in the cancer genomics field for more than 15 years at this point. So it's far from being trivial. So probably one of the first studies that tried to like systematically identify driver mutations in cancer was this Science paper in 2006. So they had a relatively limited cohort, like from today's point of view. So only 11 breast cancers, 11 colorectal cancer, and a validation cohort of 24 samples. But at that time, that was a lot for like whole exome sequencing data. And they basically tried to come up with an atlas of driver mutations back then. So what you can basically see is that the number of genes that they came up with was just huge. So it was just like a lot more than people were expecting. And when looking at those genes, a lot of those genes didn't really smell right. So there were a lot of genes involved in this catalog that were not like really involved in cancer at all. They were like more like structural genes and not really expected to be mutated in cancer. But amid all those genes, you actually saw already like those genes that now turned out to be a real driver event. They were just like contaminated amid a large number of false positive events. So early on, people actually figured out, well, there's something probably wrong the way that we try to identify cancer genes. So it's probably not okay to just like identify those genes that have the highest number of mutations, so just by the mutation counts, but you probably need to calibrate your model to the regional background mutation rate. And that was actually something that people had appreciated over time, that actually the background mutation rate has a substantial fluctuation across the cancer genome, and different like signals in the epigenomic structure of a cancer genome actually modulate its mutation rate. And so you need to intelligently integrate these like modulators of the epigenomic cancer mutation rate into your models to identify whether the number of mutations is beyond what you expect. Because if you have, for instance, like a region where you just expect a large number of mutations just by pure chance, the sheer fact that there is a lot of mutations won't really tell you that this gene is like involved in cancer. But if the same number of mutations might appear in a region that is not expected to have a large number of mutations, then actually this might actually tell you something about its relevance in cancer genome signaling. So this is like basically the first criterion, like mutational axis, but it's really important to calibrate this by the background mutation rate. And one of the first algorithms that did this was like this Lawrence paper in 2013, where they actually came up with a model to modulate the background mutation rate and integrate this into into a statistical model. And there's basically two ways that you integrate the background mutation rate. So one thing is, as I told you about, the epigenomic markers. So this model basically takes 3 markers into account of how the mutation rate might modulate over the genome. But the other criterion that turned out to be important are actually synonymous mutations. And the reason why this is because synonymous mutations are by definition all passenger mutations, because they don't change the protein structure. They will never have an effect on the cancer functions. They won't have like a driver mutation effect. Whereas non-synonymous mutations can either be driver mutations or passenger mutations. Although they change the amino acid structure of a gene, most of the non-synonymous mutations will still be passenger mutations. So what you can now do is actually compare the number of non-synonymous mutations with the number of synonymous mutations to get another approximate whether your mutational axis actually exceeds your expectation based on the background mutation rate. And there was a third criterion that we'll talk about a little bit later about like which mutation types you actually see in which genes. So for instance, if you have a C2A mutation in lung cancer, that might actually make you less excited than like a T2G mutation. All right. So they basically integrated this tool in a larger atlas in a follow-up paper and actually characterized or actually came up with the first atlas of driver mutation genes across 21 tumor types. There were a few other papers actually that make important like contributions to this field that I just quickly want to mention. So one of those papers being this Oncodrive ML method that actually like leverages the same concept of non-synonymous versus synonymous mutations, but they came up actually with another intelligence solution. Because as I told you, the non-synonymous mutations, many of them are actually turn out to be passenger mutations. But we all treat them the same in our usual statistical models. So we just count the number of non-synonymous mutations and the number of synonymous mutations. So now, as you can imagine, if there's just a lot of non-synonymous mutations that turn out to be passenger mutations, well, your model will actually be very diluted. So a better way would actually to characterize or to prioritize those non-synonymous mutations that are more likely to be driver mutations. So obviously you can do this like exactly. So you cannot like tell exactly pinpoint, okay, this non-synonymous mutation is exactly a driver mutation and this is a passenger mutation, because otherwise we would basically be done. But there is like, it turns out that there is like bioinformatics scores, and there's a ton of those. And you can actually do at least like a prediction on a like more like of a grayscale of whether this mutation is more likely to be a driver mutation or more likely to be a passenger mutation. And so by integrating these like functional scores, and that's basically an entire field for itself, but basically by integrating these functional scores into a statistical model, you can actually additionally boost the power of the original model. So in addition to what I've been telling you about like the characterization of driver mutation versus passenger mutation, there was another development in cancer genomics that was happening basically in parallel. And that was like basically mutation signatures in cancer. So basically, I've been talking a lot about how do we characterize driver mutations, but as I said initially, another important aspect is actually also to understand passenger mutations, because those always go hand in hand. So the more you understand about passenger mutations, you can actually characterize those outlier events that turn out to be driver mutation. So on the passenger mutation side, people had been coming up with this model called mutation signatures, where you basically take the trinucleotide context around a mutation and you count the number of mutations in each trinucleotide context and basically decompose this using a non-negative matrix factorization algorithm. And then come up with these like so-called mutation signatures, which correspond to different forms of DNA damage or mutational processes in the human cancer genome. So basically, it turns out that actually driver mutations tend to differ from these mutagenic processes that we described by mutational signatures. And there were actually 4 papers in the field that were kind of like hinting at this fact that actually like mutation signatures might be important for characterizing also driver mutations. So I'll just like basically mention like one of those papers where basically they basically compared like missense mutations, nonsense mutation, and silent mutation, and the latter one being by definition always passenger mutations. And they actually found that the cumulative probability of those non-silent mutations actually differed significantly from silent mutation. So the signal is not very strong, obviously, but it actually gives you an additional power boost if you combine the signal together with the mutational axis criterion that I just talked about. So that was basically the incentive of our method. So we knew that there was an additional signal out there, but it hadn't really been integrated into these models before. And it's basically the same idea or like the same principle like previous methods had worked. For instance, like this Lawrence paper, like people had been knowing, okay, like mutation rates differ across the genome, but we need to come up with a statistical model for that. And that was basically where I was standing when I started my postdoc. I knew there is something out there, but we really need to capture this and integrate this together with all this other statistical criteria that we had in the field before. That was basically the goal of building this tool called neat panning to integrate nucleotide context for driver gene identification. So why do we actually think an additional criterion was even needed? Like, why don't we just say, well, let's just use the old algorithms, just generate more data and just call it a day? Well, it turns out that the previous tools actually had been working well in some cancer types, but they hadn't been working in other cancer types very well. And it actually turned out that those cancer types that had low mutation rates, such as thyroid cancer or sarcoma, actually just counting the number of mutations works actually pretty well. So you can just like see this in the upper diagram here, where you actually see like a non-cancer gene versus a cancer gene. And a cancer gene will have more mutations because of its boost by driver mutations above the expectation. So if the expectation is already very low, then actually this will actually give you a good signal to discriminate non-cancer genes versus cancer genes. However, if your background signal is actually extraordinarily high, such as melanoma being the other extreme of the spectrum, then actually your background signal is already high, and the additional boost that you get by driver mutations is not that much that you can use for distinguishing between these two types of classes. So you'd actually need a lot of samples to distinguish these two types of scenarios. So it actually turns out that passenger mutations, they all follow like the same distribution depending on their surrounding nucleotide context. Driver mutations also tend to be in these nucleotide contexts, but in addition to this signal, they're also like guided by where functional positions exist in cancer genes. So obviously we don't know where those functional positions are exactly, because otherwise, again, we would just be done with characterizing drivers versus passenger mutations. But actually just knowing the fact that there is an additional signal that will make a deviation from the expected nucleotide context for driver mutations gives us like this boost how we can distinguish these two scenarios. So just like look at the top of this plot here. So this is like a low count scenario. And you would actually see by these additional red dots, you could actually distinguish just by counting the number of mutations very easily between the upper and the lower type of gene. But now look at this type of scenario. So you already have a lot of those mutations. And if I just gave you the number of mutations in both of those scenarios, you would actually have a hard time just distinguishing these two scenarios. But now if I just told you where those mutations are located and that the red dots are somewhere outside where I would not expect them based on the passenger mutation context, that would actually give you an additional information that you would otherwise not have interpreted in your model. So I'm not saying that by our model, we would actually come up with a better model for all cancer types, but we'd particularly come up with a better model for those cancer types where you have a high background mutation rate. And I think that's very important to consider, like that it's really depending on the cancer type, how much you can actually boost the prior models. All right, so I've basically been telling you there's two different criteria that went into our model. So mutational access, which previous people had been using, and the positions in which mutations are located. So now, obviously, the big question was like, how? Oh, sure, absolutely.
This is James. Just to, maybe you're going to get into this more, but I'm curious if you can comment a bit on sort of how in general the field is benchmarking the predictions of driver versus passenger mutations. Or
absolutely. So I would have probably done that a little bit in the discussion, but let's actually talk about this question now, because I think it is important. So first of all, the problem with our field is why it's actually very hard to benchmark the performance of your model is that you never know the ground truth, right? So if you want to do like something like a classical AUC like you would do in machine learning, you'd actually know the ground truth. The problem is here that you don't know the ground truth. So, I mean, if your model comes up with an additional gene, then this could actually be just garbage and noise, or it could be a new discovery. So that was a little bit of a problem for us. So one thing that we actually used for benchmarking our model was actually using like classical or traditional lists of known cancer genes. So there's a bunch of them, like including the like cancer gene census list, and you look whether those genes tend to be in the top hits of your significance. And the reason why we think this is a good criterion is because probably the most common drivers like TP53 and KRAS we already know. So the top significant hits of your models should be these canonical drivers. And your new discoveries, they should be, if they're new discoveries, they should not be the top significant hit, but they should be like somewhere in more like the tail of what your model sees. So if your top significant hit is something that a gene that you've never heard of, and it's more significant than TP53, then that's probably not good. So you can basically come up with, I wouldn't call it like an AUC because that's always like suggesting that you know the ground truth, but you cannot come up with something that is AUC-like. So you basically rank, are my known cancer genes among the top significant hits, and you treat every new discovery like as if it was a false positive, although it is not. And thereby you can approximate a little bit the performance of your model. So that is number one, what you can do. Number two, what you can do, and that's also what we did in this model, is to look up whether whether other models had been discovering the same gene, or whether the literature had been giving any like hint that this gene might be important. So the problem about these literature searches was always that literature is just like contaminated, right? So if you just Google a random gene, and Google or like PubMed it, and together with cancer, well, chances are pretty good that there's like just like a random paper like reporting that. So you have to be very careful about how you curate those things. But I mean, if you do this in a systematic manner, so we basically did this both for our findings, but also for completely random genes, and you just see whether your new findings come out more frequently than if you just Googled random genes together with cancer. So that was like another criterion that we used. So the third criterion that we used was looking at crystal structures. So obviously you don't have crystal structures for every gene, but for a bunch of genes you have, and you can actually see whether those mutations that your algorithm had been putting the most weight on were actually in functionally important positions that you could nominate without knowing anything about the crystal structure of the gene. So that gave us a third hint. The fourth ideal scenario would obviously be, and we haven't done that, but I think it is actually something that the cancer field has to do long-term, is obviously doing like functional experiments. And that maybe sounds like more easier said than done, because I know that the readout assay for all these functional experiments will not be the same, right? So, I mean, long-term, it's obviously, the most common readout assay that we use currently is just using like proliferation and cell survival. But I guess like a lot of those driver genes will have more nuanced effects than that. So for instance, they might compensate more the oncogenic stressor of driver mutations, et cetera, et cetera. And like those, and the readout assays that we currently use might actually not capture all the effects of those driver mutations. So that's why the fourth criterion that I've been mentioning, we haven't done, but I know that the cancer field is not doing it like to the degree that we probably should. And for a lot of genes, we just have to add it like with a question mark. So we know it's statistically significant. It kind of smells right, but we don't really know what this gene exactly does. And that's kind of like where we're standing with drivers to passenger mutations in the field. Does that answer the question a little bit?
Yeah, got it. No, it sounds like a real challenge to really clearly benchmark methods. But yeah, that's
I mean, it's a real challenge to benchmark methods, and I think it's also a real challenge to prioritize those genes that would actually make new drug targets, right? I mean, we don't just want to understand genes for the sake of understanding genes. At least I mean, of course, it's always nice to learn something new about biology. I get it, but eventually we want to like help a cancer patient, right? So I'm just asking like, how do I treat Jim based on the cancer genome? And the problem is that I have a lot. I mean, if you just like look at the patient's genome, you will find a lot of stuff based on these algorithms, but you have no idea like which of those things you should prioritize for like drug response. And so that's kind of like, I think where like there's a significant gap. And I think all these things have to like eventually like work hand in hand, like experimental people together with structural people, very into functional people, and like more statistical people like me to actually like solve cancer. Cool. Awesome. All right, so coming back a little bit to the last criterion that I mentioned. So obviously the biggest challenge of this model is to predict actually how many mutations would I just expect by chance for an individual position in the cancer genome. So I've told you a little bit about this like mutation signature model, and why don't we just use that model for predicting that? Well, it's not that this model is bad per se, but it was not developed primarily for like predicting an individual position's number of mutations. So the primary purpose for this model was to detect different forms of DNA damage. But the primary purpose of this model was not to exactly predict in that position we have that and that many number of mutations. So one thing that we actually thought was missing in this model and how we could actually boost its power is actually considering the extended nucleotide context around the mutation. So in this model, you just consider the trinucleotide context, and that's probably fine for characterizing DNA damage. But you have to understand that in our case, we don't pool all the mutations across the genome, but we actually deal with the data sparsity issue, because per position, you might just have 1 or 2 or 3 mutations, and you need to like make a score based on those mutations. So the more that you can actually learn for the individual mutation, the better your scores will be. So that's why we actually thought it will be an important part of our model to come up with a model that actually expands beyond the trinucleotide context. And actually, if you just look at mutation signatures by eye, what's been happening, for instance, in melanoma or bladder cancer or endometrial cancer, it actually turns out that the context sensitivity of those mutation signatures does not end at the trinucleotide context. So this was more like a mathematical simplicity, but it's not like biology that basically only the surrounding nucleotides affect its context sensitivity, and that's it. And that's why actually, like, to account for the entire biology, we basically expanded that model into a composite likelihood model. And basically the way that we characterize our mutation signatures, it's not necessarily by these like 96-bin models, but we can actually like come up with modes for this model where you have the entire context sensitivity. And for some of those mutation signatures, as you see, for instance, aging or smoking, it actually turns out it really ends after the trinucleotide context. So it's relatively flat signature, and there's not that much more that you can learn from the context. But for other signatures, actually, it does turn out that the context is important. And so basically, I'll skip a little bit through that, but basically what this diagram shows you on the x-axis, basically we had the, like, on the left side, we had the classical mutation signatures and the observed mutation frequencies. On the y-axis, we had basically the expanded model. If we reduce this again for trinucleotide context, and we just wanted to say, well, our model is not different from the previous model. If you restrict it to the trinucleotide context, it just expands this more generally into the expanded nucleotide context. All right, so I've talked a little bit about those criteria, why we thought this was important. a few other just like thoughts on that, like what are advantages of our model? So why can't you just like expand the bin model? So one thing is, I mean, if you want to expand for more like possibilities of nucleotide context and you just count the number of mutations, well, the number of groups or bin will just grow exponentially. So you'll just run into data sparsities that won't work. The other thing is basically at some point you'll just start to overfit things in certain nucleotide contexts. So you'll maybe have like a KRAS hotspot that are also surrounded by like exactly the same nucleotide context. So you would have one bin where you would have an extraordinary high mutation count. And this overfitting would actually kill your entire signal, because that's exactly what you don't want to do, is like overfitting into the nucleotide context and then basically kill the entire signal between driver versus passenger mutations. Plus the other thing is, and that's actually another like important concept to bear in mind, is that the classical mutation signature model had actually been looking at the more frequent groups, right? So they actually wanted to see which of those nucleotide contexts are most commonly targeted by passenger mutations. Well, we actually wanted to look at the other spectrum. We wanted to actually see which of the contexts are the least commonly targeted by passenger mutations. And that's why we actually needed a log scale model instead of a linear scale model, because if this is just like a commonly targeted nucleotide context, then, well, we're not really interested in that, but we're interested in those mutations that are actually like deviating from that. And that's why we needed to come up with a different model. So basically, just like to benchmark our model a little bit, was just like applying this to a bunch of different known cancer genes and known cancer genes, and just looking how many mutations do you have that deviate from the expected nucleotide context. And it turns out actually, yes, like cancer genes have more like mutations that deviate from the expected nucleotide context. So that's good. So that was a little bit the other benchmark validation that I've talked about. Was actually like going back to like crystal structures and to map actually those mutations that were in nucleotide context that deviated from our expectation. And it actually turns out that this model could learn, or that this model could actually predict a lot of those positions that might be functionally important, for instance, based on the protein-DNA interaction, without actually knowing anything about the nucleotide structure. And we feel that's why nucleotide contexts are actually important, because it would actually enable us to predict those positions without knowing anything about like what this protein actually does, given that we don't have crystal structures from all proteins. So basically, to summarize a little bit of how our algorithm works without getting into too much of the weeds of technical details, is basically we have a very similar problem to where people were standing initially. So initially, people had to like quantify the mutational axis above the background mutation rate. Well, that was more on a megabase scale. So we had actually the problem to quantify excess or deviations on a single nucleotide scale. So you see those problems are kind of related, but they're a little bit different. So for the megabase scale, you need to account for things like the epigenomic factors, the silent mutations, et cetera, et cetera. Whereas for the nucleotide context scale, you need to actually like account for like, you actually need to account for the like specific context around a mutation. So basically what you would do is first to come up with those signatures that you already know, which create like passenger mutation in certain contexts. And you'd basically try to associate every individual mutation with one of those processes. So to decide later whether these mutations are drivers or passengers, what you basically do is assign them to one of those mutagenic processes, and to particularly look at those mutations that deviate from this process, i.e. like those mutations where you didn't find a matching background process. And those mutations we termed unusual nucleotide contexts. And is very important to keep in mind, because I get this asked a lot, is kind of, is every mutation in an unusual nucleotide context a driver mutation? And the clear answer to that is no. It's a little bit like we talked about non-synonymous mutations versus synonymous mutations in the beginning. So all these criteria just give you a boost of telling you a little bit more on a grayscale whether they tend to be more towards drivers versus passenger mutations, but none of these criteria can exactly tell you whether an individual mutation is a driver mutation or a passenger mutation. So you just need to iterate this criteria over a lot of mutations until you have more and more of a feeling whether this gene might contain more driver or passenger mutations. So please do not treat nucleotide context as a black and white criterion. It's just more like always on a grayscale to tell you a little bit more whether this mutation tends towards passenger or driver mutations. So we applied this model eventually.
Felix, another sort of
yes.
more technical question.
Yeah.
I'm curious, is there evidence that these mutational processes are varying themselves at a megabase scale, maybe related to sort of like DNA accessibility or, for the mutagenic processes or for DNA repair? I could imagine that being another layer of complexity.
Yeah, so that's a terrific question. So the answer is probably yes, but the technical answer is, well, we don't have enough data to account for that. So what we basically did, so we were aware that there might be certain regions where just the entire process might deviate from our model. And obviously we would not want to have those things contaminate our predictions. So one thing that I didn't talk about was basically that we recalibrated the model everywhere locally, again, with synonymous mutations, and actually looked whether this process would deviate. And there were like certain genes where the entire process would just deviate too much. And we would just say, well, the model is not applicable in these processes. So yes, it's an important criterion to take into account. It's just that if you look on an individual gene, maybe you have like later, like in those genes that are like not like known driver genes, like not like TP53, but things that are maybe like less frequently mutated, you might just have 10 mutations. So obviously that will not be enough to recalibrate the entire model. So the best that you can do is just avoid false positives if it deviates, and you just know, well, sorry, in that case, we just can't apply this criterion. Does that answer the question?
Yes, thanks.
But it's a great question, and it's a thing that is important to bear in mind, because otherwise, obviously, you might actually come up with just false positives. I mean, another thing is like sequencing artifacts and like important curation of those artifacts. We can probably talk a little bit more in the discussion or offline about that, but that's also very important to keep in mind, especially if you want to do these like aggregated harmonized analyses of like large scale whole exome sequencing data. It's very important to keep in mind like what goes into this cohort and what does not go into this cohort, because an individual cohort that was just badly curated might actually just contaminate a lot of things. Cool. So we basically did this like across a large number of samples and different cancer types. And we basically come up with an atlas of those genes for every individual cancer type. So if you want to check this out, I just put the website. I mean, you can check it out in our paper, but we also made this available as a platform called cancergenes.org. So just in case you want to check that out. So it's a little bit like what we talked already about initially about how do we benchmark these things. So it's really important to keep in mind like what had been known before and what kind of like new things would we come up with. So, and obviously, ideally, the things that are always on top of those lists, they already are known before. So it's very unlikely that we'll discover a new TP53 gene. And so if you just look in general at this atlas, you can see that most of the things that are always like top ranked, that already been covered by previous databases. And then if you go down more in the list into the less frequent genes, then you'll actually see more quote unquote holds coming up. And so that's actually good. And then basically for each of those holds, we had like 4 different levels of confidence. So basically the highest confidence being that this gene is a canonical cancer gene, but hadn't been discovered by previous studies. And the lowest confidence is we couldn't even find up a single paper that would implicate in this gene. So just be careful with that. All right, talk about this platform. So also if you want to like use our tool called mute panning, we also made this available on genepattern.org. So it's like an interactive online tool where you can just like add your files via drag and drop and just like run this on your own data set. So I highly encourage you to check that out. So I'll skip that and basically just quickly come to one other aspect that I felt was important to mention and to do. So I've been talking a lot about like expanding the number of genes and gene list for every like of those mutation types. And I do think this is important, but eventually cell lines in tumor patients don't behave like linear Excel spreadsheets. They're like complex cellular mechanisms, and all of those genes interact together with each other into a pathway. So if you just focus on discovering more individual genes, well, you might always come up with additional like things that are like just mutated in 1%, but it's hard to actually evaluate how much do these things actually have in common and how do they actually like interact with each other, right? So I think it is important to keep this in mind because, for instance, if your gene is just mutated in 1%, but is in a completely different pathway than any of the genes that you had been discovering before, well, then this might not spark that much interest than if you come up with a pathway where each gene is individually mutated just in 1%, but together those genes might just be mutated as much as the canonical driver gene. So for instance, like one thing is like chromatin modification pathway. So those are the things that are just incredibly notoriously hard to detect because individually those genes are just like mutated relatively rarely. But if you basically think about that, maybe for the cancer, it doesn't really play that much of a role in which basket it puts its egg, it's kind of like, yeah, maybe there's just a spread out across different genes. And like combined, this pathway might be equally important to like a driver gene like KRAS, but we just haven't detected it that much because individual genes are just like mutated like randomly, but together this pathway is very important. So this was one of the other criteria that like our study wasn't the first one to do this. But I mean, we combined this criteria, these pathway criteria, together with like robust statistical criteria from our mute-panning model to come up with this pathway-based classification together with like statistical significance criteria. And we felt actually it was important to combine these criteria together. And so based on like combining this criteria, we could not only like quantify the contribution of individual genes to the mutational landscape, but we would actually quantify the contribution of entire pathways to the individual landscape. And it turns out that some of those pathways are just all focused on one individual gene like KRAS or TP53, but other pathways are really not focused on an individual gene. They're really spread across a lot of the different genes. And it's actually important to exactly look at those pathways because I think there is really like the most room for potential discoveries. To sum things up a little
Felix,
bit.
Maybe
sure, go ahead.
One more, that's super interesting. And I'm wondering if this question of sort of aggregating evidence of sort of functional effect from the gene level to the pathway level is like, to what degree is that analogous to, I assume there's some aggregation of evidence from the variant level to the gene level. Maybe that's not right, but if so, is that roughly analogous? But obviously in one case, you know precisely roughly like which variants are part of which gene, but the pathway is like another layer of uncertainty.
Yeah, so first of all, because of lack of time, I've obviously been oversimplifying things a lot. And I really apologize if I haven't been giving credit to a lot of people that have made important contributions. So I just want to emphasize, we're definitely not the first people who do this. There were definitely papers before which had these network pathway ideas. And there were also papers that actually used like crystal structure or like gene structure for actually driver discovery. So all of these things had just motivated us to include these ideas into our tool with mute panning. And I do think that one thing is, of course, like looking at all these criteria individually, but I think another important thing will be actually to actually combine all these criteria together, ideally in one integrative framework for the driver gene discovery. And just what I wanted to emphasize is that if you do that, you can actually maybe come up with more genes because well, if a certain pathway might actually spark more excitement, then, so like that might actually be like an additional criteria to distinguish those scenarios. But I mean, it's a highly complex field, and we could probably fill an entire talk just talking about what people had been doing before, how our model might differ a little bit from that, and how I eventually think all these things should come together. But I do think it's just important to mention that eventually the field does not just go into discovering like new like linear lists or new genes, but it's actually more like characterizing things on a pathway level. Cool. So just to wrap, or any other questions before I wrap things up, just quickly?
There was a question earlier about the extent to which sort of sequencing artifacts are accounted for. And I think you mentioned that. I don't know if there's a easy way to get into that, like a little more detail there, or if you want to maybe come back to that at the end. Up to you.
Yeah, no, sure, absolutely. So first of all, as I said, sequencing artifacts are always important to account for. So it's important to note that also other models can be completely thrown off by sequencing artifacts. There were just like a few things that we just thought is just important to keep in mind, especially for this particular tool, and to do a more rigorous like filtering of the published cohorts before, and to also benchmark them against like known filters. So we basically did this on a sample level, we did this on a cohort level, and I think all these things were just important to keep in mind, particularly not only for our tool, but like for driver discovery in general, because if you come up with things that are just mutated in 1%, I think it's even more important to take a step back and just to see, well, are these like 1% of mutations just like maybe like a sequencing artifact that just exists in an individual cohort, or might they just be like something that is like consistent across cohorts in general? Another thing is basically the publication of synonymous mutations. So I hinted to that a little bit about that the synonymous mutations are important for calibrating our models, but not all papers actually publish synonymous mutations, unfortunately. So some papers actually will only publish the non-synonymous mutations. That was like another technical challenge that we had to overcome. But all these things are more like on the technical level. They're like not necessarily like big picture or like concept wise, but I just wanted to bring this at least like to the audience's attention, that we A, have thought about this, and then B, that it is important to think about when you want to apply our tool, for instance, to your own data set, that like looking at the filters is actually very important to keep in mind. Cool. So basically, just to sum things up, basically, do we know all driver mutations today? Because a lot of people, well, there's like really like different opinions in the field, because some people say, well, we're doing all these like additional analyses with more intelligent tool, but there's like more diminishing return. And that is definitely a good answer. But I do think that we're not necessarily done. So we're definitely done with those genes that are like more than 10%. Like we won't find another KRAS gene for sure. At least like I would be surprised. Unless it's like a cancer type maybe that we haven't studied at all. There can always be surprises. Then like in the 5 to 10% range is probably most, but not all driver genes. So there might be like some surprises there. But then it really gets down to the like 2 to 5% range where, well, we do see things coming up, but if you just do a power analysis of our models, you'll actually see that our model is not powered based on the current data available to identify all of these things, particularly in those tumor types which have high background mutation rates. And then there's basically the two below field. And I do think those things can be actually important, because if you can make a therapy for just 2% of your lung cancer patients, that actually can be important. Like if you find a gene that will lead to a drug, but it's just like present in 1 or 2% of the patients, then this can actually make a functional, like a significant importance. So that's why I think basically the below 2% field is basically still the Wild West. It's largely uncharted and unexplored. I mean, we definitely do come up with some things, but our models are not powered enough. We definitely need more data or better models to do that. So I do think it really depends on which kind of like level you're looking for. But I do think for like making new drugs, even like those things that are below 2% can be actually very important, especially if you look at those things in combination with other driver genes. So basically, just to sum up things that we talk about today, is basically that most cancer genomes contain just very few driver mutations, like looking for a needle in a haystack amid like several passenger mutations. Another thing to keep in mind is basically that it has been taking us like about like 10 years ago, like basically we didn't really know that much about the landscape of driver mutation. It has been really taking us more than a decade to come up with intelligent models to figure this out. And I do think that actually, besides data generation, that the model development will be an important key contribution to figuring out the landscape of driver versus passenger mutations. So most of the driver genes that we know today have been identified by statistical algorithms. So that was a little bit what we talked about, the lack of experimental validation, which I do think is a key limitation of the field. Experimental follow-up, therefore, is needed for several driver mutations to actually confirm their driverness. So it's not enough to just say that they're significantly mutated, but eventually you really need to know what are they actually doing in cancer genome signaling to really understand what really is a driver mutation. And it really depends always on your definition of what you consider a driver versus passenger mutation. And largely, I mean, the field of driver genes has been really fueled by this like combination of large-scale data generation plus the gradual improvement of statistical algorithms. And I do think that we need to bring these concepts closer together, like I showed with these pathway analysis, to eventually come up with a holistic map of the landscape of driver and passenger mutations in cancer genomes. I'd just like to thank various people that have worked on this study, like most and foremost, of course, in my mentor's lab, Ali Van Ellen, and with a wonderful collaboration that I had with the Sunyaev lab at the Brigham and Women's Hospital. I also like to thank people at the CHIP program and the BCH, as well as the Broad Institute, and as well as my funding resources. Thanks so much again for having me. It was really great pleasure giving this talk. And yeah, looking very much forward to our discussion section later. Thank you.
I'll continue talking today about cancer genomics as well. So I'm interested in customizing deep neural network to better understand the progression in prostate cancer. As Felix illustrated earlier, that the current understanding of cancer is that it is a genetic disease that comes through the accumulation of the different somatic mutations in normal cells. So if you imagine like a certain normal cell that acquires a mutation, probably randomly or through different external factors like radiation or exposure to chemicals, this cancer cell may eventually divide and pass this mutation to the offsprings. And probably some of these newly developed cells will acquire more and more mutations. And through the accumulation of such mutations in cancer cells, that these cells will acquire new characteristics that enable these cells to grow and divide in uncontrolled way, and probably forming a lesion somewhere in the body. So I'm interested in studying the landscape of the mutations of patients, while Felix illustrated in the first talk different methods to discover driver mutations in the genomes. I'm more interested in taking all of these mutations and trying to find how they are associated with clinical outcomes. So clinical outcomes in patients, like whether their cancer is progressing over time, or whether they are responding to the treatment, or even like predicting their survival. So one problem that happens when you start associating individual mutations to clinical outcomes is that the frequency of such mutations is very low in the population. So you can imagine that the heterogeneity in the cancer population is very high, to the extent that different patients may have different, totally different mutations, while they are they have a similar phenotype. For example, they have the same cancer type. So how should we aggregate such mutations and gain like power from different locations in the genome? This is an open question. However, one natural way to do this is to think about maybe a higher level entity, biological entity, on which you can aggregate such mutations. For example, if you aggregate such mutations on the level of the genes, you may gain some more power. However, this problem still exists. So this is a representative study from our own lab that's published in Nature Genetics in 2018. And when we collected a lot of prostate cancer patients and tried to see what genes are highly mutated there, we found that there are a lot of mutated genes. And with the exception of TP53, probably, that is highly mutated in a lot of patients, most of these genes are mutated in only 1 or 2 or 3% of the population. And frankly, we didn't know exactly what to do with all of these genes, and what is the best way to prioritize these genes, or is there any way to come up with reasonable ranking of these genes to understand what's going on. So we end up with this very long tail of the genes, and we needed some way to better understand the function of these genes. If we continue with the theme of aggregating signals from different locations on the genome using higher level entities, probably one further step will be to aggregate these genes on the pathway level, as Felix indicated in his first talk. And this is like a representative study from TCGA consortium, where they proposed using these 10 pathways to study different cancer types. And you can imagine that there are so many other pathways that you can include as well and try to incorporate into your study. However, these are like one of the most representative 10 pathways in different cancer types. So frankly, the pathway enrichment analysis has been there for a while, and however, it has been used as a post hoc analysis, where you use a pathway enrichment after you do your statistical testing on the gene level, and then you try to see if your genes are clustering in a certain pathway or something like that. However, using the pathway to inform predictive models is not something that is done routinely in the cancer genomics. And in our project, we are interested in using deep learning as a predictive model to associate the genomic profile of different patients with the clinical outcomes. And as you know, deep learning is the state of the art, achieve the state of the art performance in so many fields, including computer vision, speech processing, and natural language processing. And this can be attributed to 3 major things. First, the the advances in the computational elements, including CPUs, TPUs, GPUs, and even customized hardware accelerators that can accelerate training deep learning models, and also the availability of so many huge data sets in unstructured and structured format from different sources. This enables the development of new methods and techniques for deep learning over the years. And if you project these three things on the cancer genomics field, probably we can start with the computations. I don't think right now we have a big problem with the computation and with the availability of HIPAA certified service providers. You can now run your models on the cloud without so much problems, except that you need just some money to fund your research. As long as you have enough money to buy the surface, you should be fine. So probably we are in good shape regarding the computation. Of course, there are a lot of logistic things, but at least we are in very good shape in the cancer genomics field. However, when it comes to the data, we have a very big problem. We don't have the privilege of having millions and millions of data points to train our models on. Rather, we have a very small number of samples, and even these samples are fragmented everywhere. So you can imagine all of these hospitals and healthcare facilities who are like taking care of patients. They are sequencing and storing so many patients. However, sharing these patients with these data with each other is very problematic due to so many factors. So collecting a good data set that is that can be fed into your machine learning model is a very challenging task, and putting all of these things in harmonized way and running the unified analysis on all of these data sets, taking into account the different technologies used to measure these data sets and the different computational pipelines through which these data sets were passed. So all of these are like very challenging things. And this is why one major effort in our lab is to try to solve this problem for our community. So this is the same paper that we talked about before. Is published in Nature Genetics in 2018, and we were able to collect like more than 1,000 patients. For all of these patients, we had like a whole exome sequence, and now this data is available on CPI portal, so you can go and run your own analysis, download this data, or check for the highly mutated genes, or do whatever analysis that you would like to do. So this actually facilitates the downstream analysis for so many people. And this is like a major effort in our lab that we try to solve this problem for the community. And we have very multiple similar efforts in the lab in different cancer types. When it comes to the models, if you have a predictive task that you are trying to solve, you have so many options to choose from, and the machine learning field is very rich with all of these models that you can use to solve your predictive problem. However, each one of these models come with pros and cons, and it's every one of these machine learning models has the trade-off between interpretability and accuracy. So if you use like something like simple like logistic regression, you have a very simple model that you can train very fast and you can get some results. It is very interpretable. However, the accuracy is usually less than the other models. If you go to like deep neural networks, probably you can get good predictive performance. However, the interpretability of such models is very, very painful. So our goal in this project is not only use deep learning, but also try to enhance the interpretability of such networks and try to get some insights from how the model is behaving. So for researchers in cancer genomics, it is not enough to just collect some data, do some pre-processing, and feed this into some magic model and get some performance metrics at the end. Rather, it is important to open the black box and see what's going on inside the model itself, so we can get some insights about how the model is working, and we can get like some explanations from the model itself. And this field has been very hot in the recent years, and you can think about like different techniques for interpretability that are applied to deep learning and also to other machine learning models. So for example, it is thought that in the image classification problems, the deep neural network is, it is thought that it is learning like increasing level of features. So probably the first layers will learn something like ages. The intermediate layers will learn something like parts of the input image, and maybe the last layers will learn some higher level features of the input. So visualizing such channels and filters from the neural network may give you some idea how the model is learning and what exactly the model is detecting in your image. So this may be helpful in learning how the model works, and also how to troubleshoot the model itself. Another technique may be like this is a saliency map for some image where we are trying to detect sheep in the image, and this map has like a high concentration on the sheep in the image itself. So attributing the outcome or projecting the outcome on the input space is helpful thing that can give you some idea how the model or what the model is focusing on. And there are so many other techniques like based on gradient propagation or difference propagation or different attribution methods based on attention mechanisms. So the field is moving very fast here. So we can touch upon this later. So in the next few minutes, I'll be trying to talk about these things. I'll be talking about PNet, which is like a customized neural network model that we developed and published recently. And I'll be talking about the problem that we are trying to solve using this model. And hopefully we will come to some computational results and some interpretation discoveries from interrogating the model that we developed, and even some experimental validation. Hopefully we will have some time for that. So just keep in mind that this is not a straightforward process, and usually during the development, it is like you have a lot of spirals and a lot of feedback from different stages, and one stage feeds into the other, and you go back and forth with a lot of discussions and conversations with different stakeholders, probably to make sure that we are coming up with like plausible biological insights and also superior computational performance. So I'll start first with the PNet thing. And as we know, in the deep learning language, we have some computational nodes. If you have some inputs x1 to x6 like that, you can combine them easily using some weights and get some outcome, maybe y, that's function of the inputs. And this simple model may be like a linear regression or logistic regression based on the f function. So this is a very simple model. It is very interpretable. You can go and inspect these weights, check which weight is higher, and probably the corresponding feature will be more important than the other features. If you like to have more power in the modeling, probably you need to add more layers and to come up with this nonlinear model that has multiple layers in depth, and you'll end up with this deep learning architecture where you have like multiple nodes per layer, multiple layers per model, and you have outcomes, and these layers are connected in dense way. So this is an example of a feed-forward model. However, if you have a prediction problem, there is no such guideline for the number of nodes per layer or number of layers per model, or even the kind of the connections between the layers. However, the typical practice is that you can start up with an arbitrary over-parameterized model, and you hope that you can fit the data with some help from like dropout or regularization or even drop connect. So as you can see, we have so many things that we are doing arbitrary here. So we start with arbitrary number of layers, arbitrary number of nodes per layer, and arbitrary connections, and arbitrary like dropping nodes and connections. In PNet, however, we try to add more restrictions on the architecture itself and associate some meaning to the nodes and edges in the model. So instead of having arbitrary model, we have like very structured model that is following certain, that is including our prior knowledge about the biology itself. So in the first layer, we have a number of nodes corresponding to the number of features that you are interested in modeling. In the second layer, we have a set of nodes representing a set of genes that you are interested in modeling. And the next layer, we have a set of pathways, and the connection between the genes and pathways are based on our prior knowledge that this gene is part of this pathway. The next layer, probably it is including some biological process, a higher level one that is composed of multiple pathways, and so on and so forth, until you can come up with certain outcome that you are interested in. So this seems very simple thing. However, it is powerful. So right now we have some meaning for the layers, instead of having like arbitrary layers, we have a certain number of layers corresponding to the hierarchy that you are including into our network. We have meaning for the input layers, and these are representing the feature that you are measuring. We have some meaning to the second layer. These are the genes that we are interested in, and we think they are important in modeling the process that we are modeling. We have a set of pathways and biological processes as well. And the connections as well, they have some meaning. So they are controlling the flow of the information from the input to the outcome. And all of these ages representing some relationship that is known in the biology. Well, while this looks simple, in the real scenario, we are trying to model something like that. We have access to all of the pathways involved in the reactome dataset, and we are trying to model all of these pathways. I'm not sure if the picture is clear, but you can imagine each point in this graph represents some pathway. Each connection represents a child-parent relationship between a pathway and probably a super pathway. And if you go up in the hierarchy, you have like some higher level pathways and so on and so forth, until you come up to the biological process that this pathways are part of. And you have a lot of these biological pathways as well. And in total, you have like more than 3,000 pathways involved in this modeling. So you are trying to translate all of these nodes and connections into a neural network language, and you hope that you will be able to capture something there. And of course, you can imagine that the leaf pathways or the leaf nodes in this graph are connected to the genes, and these genes are connected to different features that are measured for each gene. So it is not like very easy task. It is like involved task to translate all of these things into neural network language and transfer this abstract hierarchy into computational model that can learn something and get some results. So I'll skip this. As you may know, that the training a sparse model is more challenging than training like a dense model, and we found that we needed to do some engineering to make sure that we are getting better performance in using the sparse model. And we found that, for example, adding a classification layer after each layer in our model helps like boosting the computational performance a little bit. And these are the number of nodes per layer. And as you can see, we have decreasing number of nodes when we go deep into the model itself until we get like 26 in the last layer. This is a small number. And the first number actually is based on the number of genes that you are interested in and the number of features that you are feeding for each gene. If you sum this, all of this up, it is like almost 70,000 parameters. So technically, this is a very small number in terms of the parameters. However, it is very wide in terms of the number of nodes. So the sparse model that we developed is very, it has very small number of parameters. However, it has a lot of nodes inside the model itself. And we found that this is very helpful, and from the biological perspective, since before that we struggled into how we combine different features on the level of the genes in a systemic way. So instead of figuring this ourselves or manually weighing different features differently, the model should be able to come up with different weights for these different features to minimize the loss function that you are optimizing through the training process. We found this also very helpful since we have some meaning for the input, the outputs, and even the intermediate layers it may be helpful to go back and use some of the attribution methods and find what features are important for the outcome and what genes are affecting the outcome most, and what pathways as well. And hopefully we can come up with a path from the input to the outcome where these nodes in the middle are most important.
Haitham, can I jump in with a clarification question? So is it correct that you're using these pathway and process definitions to kind of predefine the allowed connectivity within the network?
Yes, exactly. Okay. Yeah, exactly. This is what we are doing. So we are adding constraints on the architecture itself based on the pathways, genes, and processes involved in the model that we are interested in. And all of these nodes and the edges are taken from the actome. So we are literally converting this reactome to be like functional and active computational model that can learn something. Yeah. Thanks. Totally. And probably the next slides will explain this a little bit. So this is an example of dense layer where we have like this W matrix that connects the two layers of nodes here. If you have like an X vector as an input, you can get an Y vector as an outcome by multiplying the inputs by this weight matrix. And usually this W is like a dense matrix. However, if you would like to model arbitrary sparse layer, probably you can use another matrix M here that is like represent the adjacency matrix between the input and the outcome here. And by multiplying M by W, you can get a masked new weight matrix that you can train in your training algorithm. And sometimes, for example, in the first layer, you have a pattern sparse layer where you have certain pattern for the mask itself. And this may be used to accelerate the training a little bit. Instead of like multiplying this matrix by the input, you can do some clever tricks here to accelerate the processing itself. So this is a high-level overview about the model itself. The details are published in our Nature paper last month, so you can check for the details, and I'm happy to answer questions in the discussion time. So next I'll be moving to the problem that we are trying to solve here. So in general, we are interested in understanding the cancer progression in patients, and we would like to know whether there are any genomic differences between the patients who harbor primary cancer compared to those people who have like their cancer progressed and developed metastatic cancer somewhere else. What are these genomic drivers in the, I mean, if there is any drivers, if there is any differences between these two populations, can we capture them automatically using our predictive model? And this can be measured using the computational performance if we would like to pose this problem as a classification problem, for example. And the next question probably, can we go back and ask what are these features that are different from these two populations? So we used our data that we put out for the community and ran like a secondary analysis on the same data that we published before using our PNet model. And this data is for more than 1,000 patients. Some of them, the sample was taken from the primary site, and for the others, the sample was taken from the metastatic location. And we tried to just using the genomic profile of these patients, where we have the mutation, the deletions, and the copy number amplification. We tried to model this process and solve the problem as a simple classification problem using our PNet, and compare this with other models as well. So I'll skip this. This is not very relevant here, but for the computational results, I can go quickly here. So using, yeah, I'm sorry.
Before getting into the results, is it correct that did you use like molecular features as well, like RNA expression, or no?
Well, yeah, I mean, this is a very good question. So in one of the experiments, we used gene expression, but I would say that we did not have the gene expression for all of the patients. So in the clinic, gene expression is not something that you get routinely in the clinic. Rather, you will get a lot of like whole exome sequence with mutation and copy number variations. So for the main experiment and for the results that I'll be showing soon, we used only the mutations and the copy number deletion and amplifications as input for the model itself. We tried using different data modalities like fusion and things like that. We didn't see that it is changing the performance very much. For the gene expression itself, I think it was very challenging to use the gene expression in the model, probably because of the different patch effects and things like that. But frankly, this was not the main focus of ourselves. Since we didn't have enough samples with gene expression. But this is, of course, this is like part of our future work to focus on different data models other than pure genomics data.
Yeah, I could imagine some tricky confounding factors there too, with expression from contaminating normal cells in the metastatic site and things like that. But,
sure, yeah. Yeah, I would say that gene expression is not something easy, and there are a lot of nuances come with processing gene expression, and you should be very careful when you consider gene expression as an input for your model. Yeah. Yeah, great. So yeah, I mean, using the standard training, testing, validation split, we were able to come up with good performance. And for like more than 93% of the primary patients, we were able to classify them correctly. And when we checked the area under curve and the area under precision recall curve, we found that PNET is achieving like little bit higher performance compared to the other models, including support vector machine and adaptive boosting in the random forests. I would say that the difference is not like a very big one, but at the end of the day, the performance is comparable. When we did like cross-validation, standard 5-fold cross-validation, we found even that PNET is achieving higher performance in terms of the area under curve, area under precision recall curve, and even the F1. And the variance is probably less than the other models as well. We tried to compare the PNET to the equivalent dense model, and things gets a little bit tricky here, since you need to be careful regarding defining what is the equivalent dense model here. So if you are talking about a model with this similar capacity of modeling, which means that you are comparing PNET model with a dense model that has the same number of parameters, yes, we can do this. We compare the PNET with the dense model with the same number of parameters, with increasing the number of samples, and we found that when you train both models using this large number of samples, you get similar performance. But when the number of samples start decreasing, we found that PNET actually is achieving a higher performance compared to the dense model. And you can get up to like 20% increase in the performance when you have like very small number of samples, as like 100 samples, something like that. So this was interesting. So it seemed that PNET actually is maybe helpful in the small number settings, which is like which is the case in most of the genomic studies that we have. If you are comparing the PNET, however, to a dense network with the same number of nodes, here things are even trickier. So the dense, so PNET has so many nodes actually, but we have very sparse network. We have only 70,000 parameters. For a dense network with the same number of nodes, you will end up with like I'm not sure, like 500 million parameter or something like that. So this is not like practical thing. So probably what we did here for this comparison is that we forced the first layer of the network to be sparse and all of the following layers to be dense. And this hybrid model, we just call it dense here. So the performance, the comparison of the performance is very similar. So PNET is 93. And the dense network is like 0.9. And just keep in mind that PNet has only 70,000 parameters, and this dense network had like 40 million parameters. We didn't do this for all of the sample sizes. However, we think that the comparison holds for different sample sizes, just for the sake of the time. We tried also to externally validate this network on 2 cohorts that on which the model wasn't trained. So we got like a localized data set and metastatic data set, and our model was able to capture like 80% of the metastatic patients correctly, with some errors, of course. So it seemed that the model can generalize to other data sets to some extent, and probably some more experiments need to be done here, but at least this is a relieving point that the model can generalize to other data sets. What's interesting actually that we tried to see if the model actually has been doing some errors in the prediction, and we tried to see if the model actually is doing something other than just pure mistakes, and we inspected the errors of the model, and we tried to see if there is any interesting thing in these errors, and it seemed that there is something. So for some of these patients, actually, we had some information about their biochemical recurrence, which is that the cancer will come back later in the time. Probably this can be detected through like elevated PSA levels in the patients. So we found that patients who had like high PNet score, which means that the model itself, the PNet, is saying that these patients are metastatic patients, even if they have primary cancer. It seemed that those patients had worse prognosis, which means that probably they are more susceptible. They have higher probability of developing biochemical recurrence later in their life. And on the other hand, patients who have like lower PNet score, they had like better survival, better progression-free survival. And I think this was interesting that PNet just trained on this genomic profiles of these patients, maybe learning something here that helps in stratifying patients based on their probability of developing biochemical recurrence later in their life. So this was something interesting like that we got only from inspecting the predictions of the model. So next, I'll be talking about the interpretability aspect of the model and whether we get anything from inspecting the model itself. As you know, there are so many techniques published recently, so many methods out there that you can use, different attribution methods and explanation techniques that you can use to get some insights into your model. I am not in the space of comparing all of these things. There are a lot of studies like saying that some of these models can be represented in terms of the other models, and there are other so many things in common between these techniques. But I mean, we ended up using deep lift from Stanford for different reasons, including like how easy it was and the availability of the code at that point, and the subjective evaluation of the ranking, whether these rankings make any biological sense. So these techniques actually can help you assigning scores for different features for each patient. And if you aggregate all of these scores on the population level, probably you can get some score for the feature on the population level. So for example, we did this for the inputs. We aggregated all of the important scores on the population level. And we found that this is for the first layer. I mean, the rectangular here, the bigger the one, the more important it is. So we found that the progression or the prediction of the PNet at least is driven by a lot of copy number events, especially copy number amplifications. And this is consistent with our understanding of cancer biology itself. When we did the same thing for the next layer, we found a lot of interesting genes that came up on the top of the ranking, like things like AR, TP53, PTEN, and RP1. all of these genes are interesting genes that are found in the literature. And this was like very interesting point to us, that the model is capturing known biology. And using some of the, some factorization and normalization, you can even assign some weights for these edges that are connecting the first to the second layer. So for example, here you can right away figure out that AR amplification is important, TP53 mutation, or PTEN deletion. So this actually gives you a very nice visualization of the importance of the nodes and how they are connected with each other. If you start doing this for all of the layers, you can come up with multi-level visualization of the system, where in each layer we are representing the top nodes and their connections. For the other nodes that aren't shown here, we just represented them as residual node. So you can imagine that you can start inspecting such visualization of the model and come up with your own biological hypothesis here. And you can observe the flow of the information from the input to the outcome through different layers in the model itself. And from interacting with different oncologists and different scientists, they found that this visualization is very appealing, and it may be informative in developing biological hypotheses. When we went back and checked, for example, the ranking of the genes in the second layer, or the first after the inputs, we found things like AR, TP53, PTEN, RP1. All of these are like expected to be ranked on the top. However, when you go down the list, you found some interesting genes that are not very expected in this location, like things like MDM4, VEGFR, and things like that. So things start getting interesting here. So we wonder why it is it's ranked high in the list, while it is not very known in the literature, especially in the prostate cancer realm, that these genes are like very involved in the progression of the cancer. When we inspected the individual genes, we found that certainly AR amplification is enriched in the metastatic population. TP53 mutation is enriched in the metastatic population as well, and so PTEN deletion. And we found also like a lot of like amplification events in the MDM4 in the metastatic population. And we thought that probably this is because MDM4 is related somehow to the TP53 pathways. And we found a lot of these pathways actually coming up high in the ranking in different pathway layers. So we know that TP53 is like a major tumor suppressor that actually regulates a lot of biological processes inside the cell. It regulates a lot of genes, including cell cycle genes, cell death genes, and DNA repair genes. And even interestingly, the TP53 itself is a highly regulated gene that is regulated through different mechanisms. So if you are a cancer cell that you are trying to stop TP53 from functioning, you can do so through so many ways, probably by mutating the coding region of the TP53, or through epigenetic processes like methylation or phosphorylation. Or you can do this by increasing the activity of MDM2 or MDM4 that are known to be negative regulators of TP53. So you have so many options, and it seems that the option through MDM4 is something that cancer cells are doing. So when we inspected the mutual distribution of MDM4 and other genes, including AR and TP53, we found some genes. For example, here we have like 19 patients who have amplification in MDM4 without any events in AR or TP53. We found also some patients who have like MDM4 and AR, but they don't have anything in TP53. So it seemed that to stop TP53 is not only through mutation in the TP53 itself, but you can amplify the other genes that are negatively regulating the TP53. And this was an interesting thing for us. So, we thought that we may have some interesting biology here that we would like to follow up with experimentally and see if our computational nominations have any biological meaning, or it is reasonably biological. So we went to our friends in Bilhan lab, and our friend Justin helped us with one of his nice screens that published recently. This is an ORF screen in prostate cancer cell line. And we found that a lot of our highly ranked genes are present here in this screen. For example, we found that MDM4 itself is the most highly ranked nomination that we came up with. This was very interesting. So on the y-axis, this is the z-score, and the higher, the more positive this score is, the more resistant this the cell line will be for the treatment. So this was interesting to see that MDM4 is even ranked high in this screen. So now we have two sources of evidence. The first one is coming from the computational model. The second one is coming from this this screen. And we followed up with more experiments on MDM4 individually. So for example, when we targeted this MDM4 and using CRISPR, we found the cell survival of the cancer cell lines. Different cancer cell lines are going down. And when we targeted this MDM4 using a drug as well, we found that the cancer cell lines became more sensitive, especially if this cancer cell line is TP53 wild type. So it seemed that we found something here, something that's biologically interesting, this can be targeted as well using drugs. And we did the targeting in the cell lines using CRISPR and also using drugs. And this is a very interesting thing that through the computational modeling of data that are aggregated over different times, we were able to nominate some computationally nominate some targets that we were able to validate later in the lab, and probably maybe the next step is to start clinical trials on this in this gene and see if this can like help making the cancer more sensitive to the treatment. So just for the sake of the time to summarize, we developed a PNet model, which is a sparse model, and we were able to include all of these biological information in the hierarchy of the model itself. And it seemed that it works, and computationally it was able to identify advanced cancer patients based on their genomic profiles. And we found that some of these patients with high PNet scores, they had higher probability of developing biochemical recurrence after a while. And we found that PNet discovered known biology about prostate cancer progression and also nominated new targetable gene that we were able to validate experimentally in the cancer cell lines until now. So the future thing that we are looking forward to do is of course, try to include more data modalities in the model itself, and with the hope that the more data that you feed into the model, the more accurate the model will be, and the more accurate your ranking will be. And we are also exploring different options for including the hierarchical prior knowledge, and we did this successfully using the Reactome dataset, but we are also considering other hierarchical and graph-based knowledge that we can include into our model, including PBIs and other graphs that we can get. Also, we are trying to apply PNet to solve other problems, including drug response to especially immunotherapy. And we are also trying to see if we can understand more the differences between different attribution methods and interpretation techniques as applied to the sparse network rather than the dense network.